I would like to quickly do our land acknowledgement. Fleming College respectfully acknowledges that we are situated on the Michi-Sag lands, the traditional territory covered by the Williams Treaties. Ichi Miigwech to the Michi-Saji peoples for allowing us to continue our work in your territory. Now I will pass it over to you.
Thank you, Nick. All right, so we don't have a picture of you here, Sol. I'm not sure why not, but myself and Nick are both pictured here. More prominently, my cat, because she's a lot cuter. So I'm the coordinator, as Nick mentioned, for the biotech program. Nick is student recruitment. So if you have generic questions about getting into Fleming, what's involved, what other activities, other things, maybe aside from school-specific stuff, he can definitely help you with all that kind of information as well. All right, table of contents. We'll go through kind of various areas, quick or slow, depending on how time is going. And if anyone has any questions, I can always go back and cover part of it in more detail. So we're going to look at program structure, how our program is focused, talk about the placement, because that's usually what people really want to hear about. I have some stories from students. A couple of them are linked that I can share, but if I don't have time for that, you can find all of those stories on our program page. And some of you, depending on where you are coming from, might be interested in our fast track option. So I'll talk really briefly about that and can answer more questions if that's relevant for anyone. All right, so we're going to look at the program structure. I broke it down in kind of semester 1 and 2 are similar to each other because they're very kind of introductory. So we're looking at giving you all the kind of building blocks you need to then jump into semester 3 and 4, which really start to individualize the field of biotechnology, and you get more experience in different areas with the goal of helping you kind of find what area of biotechnology do I really love. And sometimes there's lots of stuff you love, but you might also find some stuff that, you know what, I don't really like that, which is fine because it's good to know what you don't like so you avoid going in that area. So breakdown for semesters 1 and 2, the way our program is structured is we actually have 7 courses running each semester for a total of 4 semesters. And then our 5th semester is a placement. So we've condensed content over a shorter period of time just to get you out faster. So we have semester 1, Indigenous 49. My pen is working. Tablet. Oh, I can't use my tablet on this. Well, that's a bummer. Let me see if I can add it in. I don't want to do that. Okay, that's better. That's not the erase option though. So let's change the color to black because it's impossible to see other colors. We'll erase that. Okay, that should be better. So the Indigenous 49 is its introduction to Indigenous studies, which is a course that most students have to take in their first or second semester at the college. Most of the courses with the Sci starting are based in science, although some of them are not and they're still science-based. So your Sci 9 is biology, and that's part 1. And so the semester 2 is your second biology. So that's biology 2. Sci 131 is your chemistry 1 and Sci 132. So sometimes the numbering makes sense. Sometimes it doesn't, like the math 114 and math 159. They're both kind of just following each other. The numbering just doesn't make sense. The Sci 172 is scientific communication. So you're going to learn all about what kind of communication is important in science. And if we look at the state of the world today and the issues with communicating scientific information, we know it's really, really important to have effective scientific communication, and not just effective, but actually communicating the right information as well. We also have a couple of computer courses, so Comp 580 and Comp 222. The Comp 580 is just kind of an overview of your basic Word, PowerPoint, and Excel related to how you would probably use it throughout the program. Your Comp 222 is mainly focused on Excel because we're going to be using that a lot throughout the program. So we want to make sure you understand as much of that as possible. And then we're also going to be learning about a lesser known tool in Microsoft, which is Access. It's a free version of kind of a database management system, which is what many large companies use. So we're just kind of learning the ins and outs of that to model what various companies use for managing information. The other course here, so the Sci-89 is a DNA-based course. We have the Sci-90, which follows that. So we have a DNA course every semester. So it's DNA 1 into DNA 2, 3, and 4. I'll talk more about that once we've gone through all the courses. And we have a course that's kind of in the works right now. And this one is going to look at basic techniques that are really common in the field of science and regulations. A lot of the regulations relate to lab accreditation, what kind of standards we have to follow, what things have to be met, and just making you familiar with those regulations and processes. Because a lot of students, especially when they're doing their placement, depending what type of lab they're working in, they actually end up going through an accreditation process where an external body comes in and is checking that everything the lab is doing meets the standards that are required. And so it's a really good experience if you got that opportunity. So the math, and the only other course that I haven't mentioned in semester 2, Sci-93, is your physics course. It is the only physics course you have to take. I know how you feel. I'm not a fan of physics. I have nothing against physics. I have just never in my entire life had my physics aha moment where physics makes sense to me. But a lot of the physics that is being taught relates to the field of biotech. So where you might see physics in relation to the field of biotechnology and the instrumentation we're using and things like that. So we try to relate it to the program. And a lot of the math we cover kind of ties into that as well. So that's semesters 1 and 2. And the goal is just to give you kind of the basics, lab skills, as well as a knowledge base to then make semesters 3 and 4 really stand out and you get the most experience out of that because you got everything kind of hands-on skills mastered and then we can build on those hands-on skills. So semester 3 is going to be a pretty intensive semester. Why won't you let me go to the next slide? Okay, where are my controls? Down here. All right. Just when everything seems easy. Let's take off the annotation. Will that let me go to the next slide? Let me see. I'm honestly just trying to get to the next slide. And I'm not sure why it's not letting me. We have the controls. You know, the controls are always down in the corner. They don't seem to be. I'm usually much better with this, I promise. There we go. Nope, that's not what I wanted. Now I've lost all of my. We're going to. Seriously don't know why. There we go. All right, it works now. Oh my goodness, I'm so sorry. Let's turn back the annotation stuff. To the pen and actually get the eraser up so we can get rid of all this stuff. My apologies for that. Hopefully it's not as bad next time when we have to change slides again. All right, so semester 3 and semester 4, you are taking a gen ed course. So that's a student choice gen ed course. There's a lot of really neat gen ed courses. One of them that I would recommend, just because of how prevalent AI is becoming, is there's one based on AI and really teaches you about um, what AI can do, what it can't do, how you can get caught up in various things related to AI. So I'm going to strongly recommend that one, but it's up to you. The Sci 91, that's your DNA 3. So that's continued from semester 1 into semester 2, semester 3. And then Sci 92 is your DNA 4. And I like to use Roman numerals. I'm not sure why. Sci 94 is microbiology. Sci 95 is molecular biology. This is it's called a business class, but it's more professional practice. So the goal of this course, because we have a placement in semester 5, the goal of the professional practice course is to make sure that everybody has a resume put together, knows how to write a cover letter, has practice at that. There's also practice doing interviews, a whole slew of things to help you practice and get ready for your placement, because a lot of the placements will ask students to interview. Some of them just want to see your resume. Some want to see a cover letter and a resume. Some like to do an in-person interview. Some are okay with a virtual interview. And there are a rare few that will actually have you come on site and do a competency test. So it's kind of like, okay, we see you're good at this, you've got good grades, you're a great fit with the people in the lab, but we want to see your actual pipetting skills or this skills. We actually want to watch you do things. So that professional practice is kind of saying, okay, these are all the possible things you might run into, and let's prepare you for that as best as we possibly can. And some of our other courses, like the Sci 90 and the 91 and the 92, so semesters 2, 3, and 4, we actually, as part of some of your testing, we put in little competency tests. So we start off with a really small one in semester 2, we make it a little bit bigger in semester 3, and then it's a bit more involved in semester 4, and this is to give you that experience if you show up in a lab and they're like, okay, you need to calculate this, and then I want to see you pipette this, and then I want you to make this and do this with it. So if you had to do those things, we've helped you practice for that. We have an automation 68 and an automation 69, and this is all about lab automation. So this is lab automation part 1 and lab automation part 2. This is to teach you about the instruments that could be found in some of the labs you end up in and the level of automation that can be incorporated into those instruments. So liquid handlers are a big thing that a lot of labs use, some of them quite extensively, some of them on a smaller scale, but that's one of the things. So you learn about these instruments, you learn about programming these instruments on a variety of things around working with these instruments. So both of those courses focus on that. And then Sci 100 is a really long title, Lab Operating Standards. Operating Standards. And it's part 1. And then the Sci 100 is part 2. The basis of a lot of what you're covering in the lab operating standards is kind of a continuation from that course that we're developing for semester 2, where you start learning about regulations and accreditations. These two courses build on that. So this is about quality control, QC, and quality assurance. So these, in general, the topic itself is boring because it's just, are you following the rules? What rules are laid out? How are we checking that? How often do we need to check? What kind of standards do we need to hold you up to make sure people are running and operating properly? So it's a lot of information that is often very dry, but it's also an area that is pretty big in terms of job availability. There's a lot of opportunity to grow in this area, and it's a key part to any company, but you don't notice how much of a key part it is until something bad happens. And usually when something bad happens, let's say there's a recall that's happened in some sort of processing plant, whether it's making baby food, that was a big thing for a while. Pop-Tarts, not Pop-Tarts, Pizza Pops was one thing. We had recently bought some, so I was going to the freezer to check the UPC code if they were okay. But it's like E. coli or salmonella or things like that that were discovered. And that's where you go back and you're like, okay, what quality control and what quality assurance processes do we have in place? How did this happen? And so the stories that happen as a result of failures of quality control and quality assurance are pretty big deal. But when you actually look at it as part of a business plan, it's not always something that people take as seriously as they should. So we'll learn a lot about that. And then semester 4, so we have a bit of a continuation for the Sci 91 into 92, so the DNA 3 into DNA 4. The lab automation continues and the lab operating standards also continue. But then we introduce some new topics. So the Sci 96 is biochemistry. And so that's kind of taking off from the point where molecular biology stopped. Biochemistry picks up. The Sci 102 course is about pharmacology and toxicology, and it takes a forensic toxicology approach to the toxicology side of things. And then the Sci 146 is all about analytical chemistry. So that analytical chemistry is going to teach you about how do you test for the presence of things? How do you measure for the presence of things? What kind of instruments can you use, techniques, tools, kits to detect things? So a lot of analytical chemistry is used in water, whether it's water testing, water treatment plants, wastewater treatment plants, a variety of things. So the goal here is that semester 3 and 4 really help you to explore a lot of the different areas that fall under the umbrella of biotech, because biotech is such a huge umbrella. And we don't cover every possible thing. And so sometimes that leads students to a semester 5 where they're like, okay, I really like this, this, and this, but what if I did a placement here which uses a variety of skills that I've learned, but in an area that I haven't actually taken any courses in? And that's always an option. And sometimes that doesn't necessarily happen in placement, but it can happen after your placement when you go to get a job. And we'll see a little bit of this. I have one student I'll talk about where they have gotten to since graduating from the program, but that's just a breakdown of what we cover in the semesters and why we cover it that way. Okay, let's see if we can more seamlessly move through. Okay, I just have to erase it in between. All right, just some statistics, just to give us a break from all the information. When you were in lab, so over semesters 1, 2, 3, and 4, you're getting close to, when we total everything together, 500 hours in lab, so learning hands-on skills to prepare you for the field of biotechnology. It's going to start off slow in semester 1 because we're kind of like, oh, these are the basics. This is how you hold this. This is how, like, really basic stuff. But after you learn the basics, then you're just thrown in. It's like, okay, now you need to use this every time we have lab. And so some of the things, pipettes, are going to become your best friend. You're going to be using them all the time. So those 500 hours are meant to give you those lab skills. So when a placement or a job says, okay, well, what have you done in lab? You can talk about the things you've done in lab. And so that's the first 4 semesters. And then your placement can be up to 600 hours. There's a minimum requirement. Because we're an advanced diploma, there's an hour requirement that must be met for lab hours. And so your in-class time in semesters 1 to 4 gets you close to 500. Your placement gets you the remainder of them, and it just depends on what availability you have to give for hours, what the placement is looking for. So we kind of find an in-between that works best for both. And the more experience you can get at placement, the better. Now that 600-hour placement happens entirely in semester 5. That is all you are doing in semester 5. You have one course. It's called FLPL 97. That's our course code for it. It's your field placement. That's all you're doing. So you don't have any other courses. You have to post once a week about what you're doing. You have to complete some surveys at the midpoint and the end. Your placement supervisor completes some surveys. And as long as you meet those basic requirements, your placement is complete. We really want you focusing on your placement. It's like a job for 4 months. And the way we've structured our program, the semesters and the placement, you get your diploma in just 2.5 years. So the condensed format allows your completion sooner than you would if it was spread out over a 3-year period. But completing it in the 2.5 years, it's still equivalent to a 3-year advanced diploma, which is a relevant feature for international students. Right, figured it out. There we go. All right, program focus. What is our program focus? Talked about this already. Hands-on. We want you in the lab. We want you doing the things. Lab-based skill set. So you're going to develop skills that somebody would use in the lab. And just because you're doing skills in a microbiology course or a molecular biology course doesn't mean you can't use those exact same skills in a different type of lab. And a big part, and this is going to be true no matter where you end up in our program, another program, whatever it is, troubleshooting and problem solving skills. Those are so important, especially in the area of science. Some labs are going to be very formalized. It's like you have to follow this process and it's the same process every time because they have to follow standards and they're accredited. So that doesn't offer much room for troubleshooting and problem solving because you usually have to follow a very specific path. But some people end up in more academic or research-based labs, and that's where those skills are definitely going to serve you. But coming up with new techniques or new ways to do something or assess something, those are always areas where troubleshooting and problem solving can come. All right. Let's get rid of those. So the hands-on hours, as I mentioned, 500 or almost 500 hours that you will get in the lab. And then your placement is going to add up to another 600 hours of that. And not only do you get more hands-on time and lab skill set building with your placement, but you're also going to be able to say, I have 4 months of work experience that you completed as part of your diploma. So you're kind of like, I've got the education and I got a bit of work experience. Now I should be a more suitable candidate for somebody who says, okay, I want you to have this education and I want you to have some experience. You've got both of those things in the advanced diploma. And the lab-based skill set, as I mentioned, you're going to start with the basics. How do things work? A little bit of theory behind it, holding things, learning the parts, and then you're going to start using them to do the things. A good example that I can think of for this, your Sci 89 course. So that's your the official name for it, Forensic DNA Applications 1. So the first one, you're just learning for the first couple of weeks. I think the first 4 weeks is just we're reviewing basics from math, how to do calculations, how to hold a pipette, how to add a pipette tip, how to take the pipette tip off, how to read the volumes on it. Like really basic stuff, but we're giving you a lot of time to get really familiar with it because as soon as we finish that, the next thing you're going to do is you're going to start a DNA extraction. And you actually learn how to do 3 different DNA extractions. And then you learn techniques to quantify and qualify the results of each of those DNA extractions so that you can then see, okay, which one was best? How did I do? And you're practicing your skills this whole time. So you're learning how to do stuff and you're practicing your skills. And then the next DNA course, you're going to repeat some of that stuff. So we already assume it's like, you know how to use a pipette, you know how to do the math, you're going to go right in. And you actually get to extract some of your own DNA. So that will be something that you get to do a little bit of profiling at a couple of loci. And then semester 3, you actually get to do, we call it a mystery meat lab for your DNA course. So you get a mysterious piece of meat and you have to identify the animal that it came from. And you also get to work with some plant DNA in that semester as well. And all of that builds us up towards semester 4. So we're actually in semester 4 right now for one group of our students. And they are in the process. Most of them have finished extracting their DNA. And it's actually, again, it's their own DNA. They either did cheek cells or they spit into a column. If you've ever done any of the Ancestry or the 23andMe stuff, the spit column, we use the same thing. They're extracting their own DNA, and we're actually going to subject it to a DNA profile. So very similar to what any forensic or policing service would do if they wanted to get your profile. We will have students doing that, and they'll be able to get their very own profile at the end of the semester, and they'll have to present on that. So it all builds you up and you see all the things that you could be doing with DNA. And this doesn't just apply to humans, it applies to animals. So wildlife forensics is a huge field in various parts of Canada and around the world. We have the Ministry of Natural Resources here, something called eDNA, and the E stands for environmental, is becoming a lot more popular, especially within the ministry. And the idea would be, let's look at a lake or a pond. And you're like, I wonder what kind of fish species are in there. So the idea is you would sample water and you would be looking at the environmental DNA in that water. So you don't actually have to catch a fish. You don't even have to know if that fish was there, but you can sample some of the water, extract DNA from that, and look for specific things. And they actually have used this quite successfully in a number of places to detect invasive species. So when you think forensics, you don't want to just limit yourself to the human side of things. There's also environmental, and then you get into the health side. So not really forensics, but the genetics of health and also the genetics of bacteria and how we study that and how that relates to health or other areas. Like it can balloon into so many different areas. And then the troubleshooting and problem solving, no matter what, these are going to be important skills. So it's the process of looking at a problem, thinking through it, coming up with a plan, being very systematic, making sure you test one thing at a time. It doesn't mean you're always going to have the right answer, but it's important to always be thinking about it. So we will always challenge you. If you come up and say, okay, this didn't work out, what did I do wrong? I'm going to ask you what you think you did wrong. It's like, back it up. What did work and what point did it stop working and what might have caused that, to really think about it and to understand it yourself. Let's try this again. All right, placement. So I'm going to erase all this. So placement, semester 5, it's usually what students want to hear the most about and talk about the most. Students come into the program and have a really specific idea sometimes what they want to do. And sometimes at the end, that's still what they want to do. So that happens. But I'd say more often than not, what happens is students start the program, realize what options they have available to them, what really falls under the kind of umbrella of biotechnology, and what they wanted to do has kind of morphed into something else. It might still involve what they initially wanted to do, but has kind of gone off on a different direction. So the idea of placement is you've kind of found an area that is really of interest to you, and it's an opportunity for you to explore that area to see if you might want a job or a career in this area. It also gives you an opportunity, as I mentioned, you have 4 months of industry experience and networking. Networking is a huge opportunity and a huge thing we always recommend students to do on placement. Students are like, okay, well, if I finish my work early, can I leave early? I'm like, you know, it would be a good idea, not leaving early and asking if anybody else needs help. Or maybe there's a department you're not allowed to do stuff in, because that sometimes happens based on the nature of the work or the level of experience that's required or standards that have to be met, whatever that might be, but maybe you can shadow them. So, hey, I never got to do this myself, but I shadowed this. I got to see what goes into it. So there is still some learning that happens there. The placement process, how it works, it's student driven. We don't actually start working with a group of students on placement until they've reached semester 3. We want you to get through the beginning part, have a chance to really start thinking about things, and then start experiencing semester 3 before you start thinking. And that process works into semester 4 as well. And so it's student driven, meaning you might have specific geographic limitations. It's like, I live in Peterborough, my family's here, I'm going to stay with them while I'm completing my placement. I need to be in the Peterborough area. And so we will look for opportunities. You will look for opportunities in the Peterborough area. And if you can drive, how far out are you willing to drive for your placement? So we look at those kinds of things. Other people aren't from Peterborough, they want to move back home. And so back home could be anywhere else in Ontario. Some people who may not be from Ontario move back to wherever they were from in Canada, and you can do your placement anywhere in Canada. In theory, you can do your placement anywhere in the world. There might be different rules for international students, so I'm not going to say that that applies to everybody. The only challenge that happens when you leave Canada is having the appropriate visas. And that's something you would need to figure out on your own to ensure that you can actually do a placement there and it's not, nothing bad will happen to you if you don't have the appropriate paperwork. So students pick based on what type of placement they'd like to do and their geographic area, if there are any limitations. Now, sometimes if you have a geographic limitation, that could potentially limit your placement opportunities, but we work as much as we can with students to make the best opportunity possible. So students really drive this process. They do research. They find a list of companies. It's like, hey, this is a really cool company. They're right nearby. I could drive there if I needed to for placement. And they give us as faculty, so we have 4 full-time faculty who would work on this. You give us the list and we start reaching out to placements. So we do that for you so you don't have to. You just make the list so we have that as our starting point. And then we also have connections. So some placements always come back to us and say, hey, we'd love to have a student again. And so we post those opportunities as well. And sometimes a place a student really wants to do their placement and a place that wants a student, that works out together. One place, not always, but often takes placement students is the Toronto Zoo. A lot of the work that they do at the zoo relates to not so much DNA work, but a lot of it is related to cell work and hormone studies, because a big part of what they do at the zoo is fertility testing and programs like that to ensure that they can continue a species and can monitor reproduction and do all the right things to maintain a species. So that is, just as an example, a place where students have done their placement in the past. And there's lots. So let's see. I think the next thing we're talking about is stories from students. And some of this will tie into, erase that, what we've already seen. I do see that we're getting close to our 45, so I probably won't play any of these videos. This one here is linked to a video, but you can also find it on our program page. So if you just type like BTF Fleming College, it'll take you right to our program page. Tons of information on there, but if you scroll all the way to the bottom, there are 4 videos there. This video is Sarah Donald's, who did her placement. You can sort of see her badge here. She did her placement at OICR, so that's the Ontario Institute of Cancer Research. And so the video just kind of talks about her story. The image on the right is another link. It's another video on our program page, so you can see that there. That's Madison. And she actually has a very personal story that kind of led to why she came to the biotech program and why she wanted to pursue the type of placement that she pursued. And it relates to her own kind of health struggles. So it's a very inspirational video that she did. And then the image on the left is a grad of the program, Mary Jo Prakash. And she is actually working her, you can see she made this AI image of herself. She's like, I kind of put in all the key details of what she was doing in the lab. So she works for a company, Cooper Surgical. She actually just told me that there's another grad of our program working there as well. And this is actually a fertility clinic. So that's what she's working in. So none of what we have in any of our program stuff had anything to do with fertility clinics, but a lot of the skills, the knowledge, everything you're learning can lead you to that type of job. So lots of opportunities there. And I think the other images. So the image here is a bunch of students graduating. There's a bunch of faculty there. So every June, they have graduation ceremonies. We do pictures. A while ago, it's been a couple of years now, they used to do it at the wellness center that was just like a block away from the college. And then we'd come back to the college and do pictures and all that stuff. So that's what this picture is from. More recently, we do it kind of downtownish Peterborough, just outside of downtown at the Memorial Center. The image on the right here was taken, this is in one of our main labs. They call it the chem lab just because it has all of the setup. Pardon me, to do a lot of chemistry stuff. And this is actually after one of our open houses. And we have a lot of students that come and volunteer for the open houses. The students are the ones that really tell other students or future students about the program. And actually, this gentleman here in the white t-shirt, Matt, he completed our program. I'm drawing a complete blank where he did his placement, so I apologize. But he actually went to the University of Guelph, and we have a agreement with them. So he completed our program and then did an additional, I think, 2.5 years at the University of Guelph to get his degree. And he's actually now working at the Center of Forensic Science in Toronto, working in their labs. And thank you, Nick, our open house, April 11th. It's usually, I think, from 10 a.m. to 1 p.m., but usually people, I think they start opening earlier than that, so you can register earlier. We try to get to the lab earlier as well. And then the image on the bottom is a bunch of students. They're not all of our students. Some of them are students, and they're actually at the CAWT, the Center for Alternative Waste Treatment, which is actually located at Fleming's Frost campus. And they, several students we have every semester, usually students doing their placement at the Center for Alternative Wastewater Treatment, and there's lots of things that they can do there. All right, fast track option. I think that's the last thing. So anybody who has already completed a degree, and we have a couple of degrees where we kind of already worked out all the details, and it's like, yes, enough of the stuff matches, you're good. It's kind of an automatic. We would look at any kind of science-based degree from any university. Again, international students, it doesn't always work out. I think we're still working out the details for that. So I'm not going to say for sure that it's an option yet that is open for international students. But the idea is, I talked all about semesters 1, 2, 3, 4, and 5. As a fast track student, as somebody who has already completed your degree, you would come in and start in semester 3 of the program. And so because you're joining us halfway through, you're not going to gain as many hands-on hours on campus. It's about half, 250 hours instead of the 500, but that makes sense because you're not attending the first 2 semesters. But you're still going to get placement, and you will still have gotten the time you spent in lab during university, counting towards kind of your total number of hours. But it's kind of like you're just bulking up your degree with additional lab kind of hands-on hours, and then that industry placement, which gets you in, and sometimes is a great network opportunity. And sometimes students are like, you know what, I don't really care what I do for my placement, but it's my goal to move to, and sometimes they want to move to the States, the UK, Australia. Sometimes they want to move somewhere. And I'm like, what if you looked at companies that have locations worldwide? And maybe that's an opportunity. You do your placement there and you're going to get access to kind of some of their internal applications and things like that, that you could maybe use that as your way to get to the country that you want to go to. So it's not necessarily focusing on what you really want to do, but kind of helps you fulfill another kind of part of your life dream that isn't necessarily career-based. All right, I think that's it. I think I'm done talking. I could talk forever about this. But now, this is our final slide. And I've totally lost, there we go. I lost people's faces for a while. So I think I'm probably okay to stop sharing my screen now. Is that good? Okay, I'll stop sharing my screen.
That's great. And if anybody has any questions, they can, oh, there's displacement pay students, salaries or allowances. Which bachelor programs?
Let me open up the chat. I just want to make sure I can see the. All right, the placements. So the nature of our program, the nature of what is defined as a placement as part of our program, it is by definition unpaid. However, some placements are paid. So some companies can pay their students. And if you were to find a placement that is paid, we have absolutely no problem with that. So we don't stop it from happening. It's just by default, placements are unpaid. And what bachelor programs can I progress this diploma to if I want further studies in science, healthcare in Canada or later? Can I progress this to medical lab technology? Yes. I'm trying to think. I know students that have completed our program and went on to, is it called Ontario Tech now? Yoit used to be Yoit and now it's Ontario Tech. So I'm pretty sure we did in the past and we might still. I'm just not as up to date on all of the agreements because there's so many and they keep changing. But we have had students, I know one student that completed our program, he did his placement and then he went to Ontario Tech when it was still U8 and he got his medical lab. I can't remember if it was an MLA or an MLT. But then he since went on, he's actually working at the lung transplant biobank at the University Health Network. And so he kind of took that in a different direction. We do have a lot of students that end up in various like Dynacare and Life Labs on their genetic or toxicology sides as well. Some of them have continued and gotten their medical lab technologists. Some of them have not. And so it's just a different level of what you can do in the lab. And it's usually just people with a medical lab technology or technologists can sign off on things, whereas somebody who doesn't can only go up to a certain point with what they do and they need sign off from somebody else. So kind of like a pharmacy technician can do a lot of the work, but the pharmacist is the one that has to sign off on the final prescription. So that kind of thing. And we have had students, there's a Dynacare location. It used to be in Bowmanville, and I think it's in Brampton now. I'm pretty sure it's They just moved their location. So it used to be really close to Fleming, which was great, but then they moved, and so it does a lot of that work. And you don't need a medical lab to be a medical lab. You don't have to be accredited in that position. And then I just want to see what bachelor programs can I cross. So yes, a lot of students have gone into, so some students go to Trent and there's a Trent forensic degree that you can do, biology. I know a lot of students have also gone to Guelph and there are a variety of degrees there as well. A lot of the universities will come to the college or have speakers or reps at the college and you can go talk to them. So usually I get that information as the coordinator and I would share it out to all the students. It's like, hey, somebody from Guelph is going to be here on Monday. Somebody from Trent is going to be here on Tuesday. So if you're thinking about, yeah, I'd like to do university next, you can go talk to them and they can tell you, oh, I'm the person to talk to, or they can tell you the right person to talk to for that. There have been students in previous years who have done placement for their education in forensic toxicology. Not in a while. I have to look back. There was a lab in somewhere out west. But I'm blanking. It was a large company and they had locations all over the place. And I'd have to go through my email. But that was the only one that I can think of where it was really forensic toxicology. I think I think it was like equine, like horse toxicology and other things like that, but I'd have to go back through my email. It's been a really long time since forensic toxicology has come up. And if you're looking at something like the Center for Forensic Science as an example, because they have multiple locations and different locations deal with different. So, like the one in Toronto does a lot of biological samples and DNA stuff, whereas other locations are like paint, arson investigations. So, like what kind of accelerant was used. So, that's maybe more the forensic toxicology side of things. But we ask students to do that research, like find places, like where would be a good place to do forensic toxicology? Because I'm definitely not going to know all of them. And students often come with like, I never knew that place existed. That's such a really, like, it's a cool place. Let's see if we can get a placement there. So we definitely try to support students with that. And then if you're looking for further education for forensic toxicology, I mean, that would have to be a quick kind of Google search. I'd say probably U of T would probably be a good place to look for something like that, just based on what they do. Um, but you can look on kind of the health science side of things. So, Sick Kids in Toronto, University Health Network in Toronto, a lot of the hospitals that also have research associated with them. So, Sick Kids is great because they have a huge, like half of it is clinical testing. So, all the patients get tested on the clinical side. And then the other half is the research. So, a lot of patient samples that have been approved to like have more testing done, they do experiments and they do testing with those samples to learn more about certain diseases or disorders, come up with better testing protocols or kits or things like that to improve results, patient outcomes, things like that. So you might find kind of forensic toxicology in that area instead of kind of the more forensic or policing side of things. So there's a lot you could potentially explore there in the area of forensic toxicology. Nick, do you have this information? You're muted right now, just so you know.
That's a tough one. It depends on where you end up landing and what you can negotiate. Again, we don't guarantee employment out of a program, and it's like we can tell you
I
career opportunities, but not necessarily salary range.
Yeah, the salary range is tough. Just to be clear, biotechnology is not like an accredited program. So somebody might say biotechnician, biotechnologist, like there's no distinction between those two. There's no accreditation to it. So one doesn't, and different companies have different ways of like labeling things. I had posted a while ago, Myopalette, which is a company I think in Toronto or like the GTA area. We've had students do placements there. They've since been hired afterwards. And the posting, I think the range was saying the starting salary was 55 to 60,000 as the starting salary for what they were looking for. And I think that was just a generic technician role. To give you it's just really hard to know what that is. I think it'll be easier now because didn't the rules just start where they actually have to post salaries now with jobs? So I think it'll be a little bit easier to start to collect that information, but unless we actually go to students and say, "Hey, how much are you making?" we can't really collect that information. So it's a little bit challenging there. But Nick is right. Where you end up, so the actual geographic location is going to pay more if the cost of living is more. What level you are. So if you already have a university degree, the chances of you starting at a slightly higher salary are better. Um, Negotiation is definitely part of it. Sometimes there's an opportunity to negotiate, sometimes there isn't. It just depends. And I think my big thing with placements is usually you try, if you can, to get a job with your placement afterwards. Sometimes students don't want that. Sometimes students do the placement and they're like, this isn't actually what I want to do. I kind of had a vision of what I thought it would be. I tried it out. It's not what I thought it would be. Or it's just the company isn't the right fit for you. Or the geographic location doesn't turn out to be where you wanted to live. So those things change as well. But um, I don't know if that gives you enough of an answer. It wasn't a great answer.
I just want to add that there are so many areas too. I'm always fascinated with this program. When you look at the employment opportunities, what you can do, it's so big that it's hard to say a salary, because you can go into the food industry also, like so many things, that quality control, it's so many things that you can do and so many industries you can go to, that is just hard to tell. But thank you for trying to both for trying to answer that one.
Does anyone have any other questions, or shall we wrap up this evening?
I don't think we have any more questions, new messages. Let me just check.
Doesn't look.
No, I dropped the ask us at flemingcollege.ca email. Mandy, let me know if you want me to drop yours on the chat as well.
It's always great to have the program coordinator's email for really specific program questions, because we end up just sharing the email anyways.
I'm trying to find, and it's easier if you email me this too, if you're like, I was trying to find, because we have a LinkedIn page, and we use our LinkedIn page to share all sorts of job opportunities and all that stuff. I'm trying to find a good link to it, but I don't have a good one. So I will, like, if you want to know, like, access to that, I can share it with you so that you can follow it. But we sometimes have placements or people that we're connected with reach out and say, hey, we have a job posting. Can you share this to your students? And so we will share all possible job postings that we can there. So that's a good place. It's also a good place to connect with previous grads from the program and see where they're working. And sometimes that's your network into kind of another opportunity. It's like, hey, I just applied for this job. I just finished the biotech program. Can you tell me anything about it? Like, it might be a good way to kind of network and get a little bit of a leg up sometimes, because you're alumni of the same program.
Yeah, definitely a good idea.
I think that looks like it's everything, gang. So at this point, I guess I will stop the meeting, and thanks for everybody for coming. Thank you to our program coordinator for participating this evening. Thank you to Saul for the international aspect too. It's great to see everybody. Thanks so much. And don't forget that openness is April 11th.
Thanks, Nick.
Thanks.
Thank you, Nick. Thank you, everybody, for your interest in Fleming. See you soon.